rolling here.
Do you know who Parker Housewright is here?
Did you get that email too?
Okay, because I thought this was really weird.
It looked, because the grammar was a little funky, and I was like, okay.
We have been invaded several times by spam emails with stuff like that.
The email I got is from a Parker Daniel Housewright, and it says, "Hello, I'm staff in the college at Dr. Cheryl Link for students who might be interested in a paid part-time personal assistant position. Job to make up to $500 weekly. He offers pay $500. Please contact John Sands 28@outlook.com for more information. Remember to email him with your private email, not your school email when applying. This is a work from home position. Have a good day."
That's, yeah.
But it's
Yeah.
from a Walter State email address, so I'm like, what?
Right, they've had stuff come out from our dean asking us about this, or other staff members. So every now and then we seem to get penetrated in the system. So generally we just try to ignore it. No one's gonna, any kind of position would be posted to everybody.
Not sending an email.
Not send us an email, and so you would see it. It would go out to the whole world. So we have to, by law, they have to do that. So
Don't fall for it,
So-
basically.
yeah. So once.
Yeah, it's like I work like 40 hours a week and I don't like those.
Mhm. So, okay, we're going to go ahead and get started now. I had thought I wanted to do the final exam review today for you, but I've got some complications at home. I posted the video from last December, and it would be exactly the same thing I would have done today. So with the exact same questions. And this is for the AP2 final exam. This is the same one I used last fall, and the AP2 class last fall did very well. It didn't give you the answers, but it did walk you through everything you need to remember. So those of you that had AP1, most people did really well in the AP1 final. I was very pleased last fall. It's on the same nature as that. So if there is a groundswell of enthusiasm for me to do a new review, I'll be happy to think about a time when we can do it. But right now, I've already posted the video. So if you want more, let me know. I'll be glad to do something else. So anyway, what we're going to do today. Oh, thank you. Yeah. Oh, somebody would have noticed eventually. So
They can't see it. You're just sharing the screen with them.
I haven't even shared anything
Oh, you
yet.
ain't shared anything yet.
So all they're hearing is us talk.
Yeah.
So I was getting ready to share the screen. So okay, now let's go ahead and open this up. Okay. Now, what we're going to do today is take a look at the immune system. I'm going to go through the non-specific immune system first, and then we're going to start into the specific immune system and try to give you a good idea of how that works, how the specific immune system works, and how it relates to, we've been hearing a lot about immunity for the past 2 years for some strange reason. I don't know what for, but we're going to give you a good idea of how vaccinations work and things like that. So let me get this out of the way. And so now our immune system, it protects us, and we have three parts to it. The first part is the non-specific system, and that is the things that just protect us in general, from the random pathogen. A pathogen is a disease-causing agent. It can be a virus, it can be a bacteria, it can be a fungus, it can be, a bacteria that produces a toxin, for example. That's a pathogen. Now, we have two very unique non-specific defense mechanisms. The first one is our skin. Our skin and our mucous membranes. Our skin's waterproof, essentially. It's impenetrable. Bacteria can't get through, okay? I don't feel like trying to say that word today. Bacteria can't get through intact skin. If the skin is torn, if there's a lesion in the skin, it's an entry point. So our skin is a perfect barrier. The sweat on our skin has antibacterial properties. Our mucous membranes also protect us. And stuff gets trapped in saliva and we swallow it and it's destroyed in our stomach by stomach acids. Things that we inhale are trapped in mucus and brought to the same location. So we really work very hard to protect us from any kind of pathogen. Now, a cut in the skin, the barrier is broken, so pathogens can get in. If we eat something, drink something, touch something, and then rub an opening in our bodies, we can allow bacteria or some pathogen to get in. A very common place of entry is our eyeballs, because we rub our face and then we rub our eyes, or we touch something and we rub our eyes. And so now we've provided a mechanism to get in there. You know, we all touch our face. You're touching your face
All the time.
all the time. I just rub my nose and you're touching your mouth right now, you know. So we do this all the time. We can't avoid it. It's always fun to do this. When I start this lecture and just pick on somebody who's been doing and you're rubbing your eye, there you go. So, yeah. So we cannot help doing that kind of.
Talking about it makes me want to do it more.
Of course, I'm like dying to rub my eyeballs and scratch my nose, So, but that is our first line of defense, because pathogens can't get through our skin. They can be on the surface of our skin, but our skin is essentially waterproof, and things are they don't burrow into our skin. Viruses and bacteria don't work like that. Now, if there's any lesion, any tear in the surface, then we're exposed. But generally, we are protected. And we also have our own level of bacteria living on our skin that is our bacteria. It is the stuff that it's what we call the indigenous bacteria. It's the stuff that lives there. It protects us. It's sort of like our pets, if you will. It is the bacteria that fills up all the spaces on our skin so foreign bacteria can't get in there. And that's what happened. The problem with that is that if we wash our hands with an antibacterial soap, we kill everything. And it stays dead for a long time, long enough for any kind of, as we say, an opportunistic pathogen to find a home on the surface of our skin. And it can stay there long enough to find a way to get into our bodies. Whereas if we just use plain ordinary old soap and water and or a sanitizer, with alcohol, it only kills things for about 20 minutes. And our own bacteria can repopulate quickly. The antibacterial soaps, as I said, it's like a dead zone for hours. And it gives with our own bacteria missing in action, our pathogens, the pathogens we may encounter can eventually get on there first. Yeah.
How long does it take for everything to regenerate after we wash it with the?
Hours, from what I understand, hours, like 4 or 5 hours, because you create a dead zone, it lasts on there. I mean, it does its job very well, but alcohol.
Does it immediately start regenerating, or does it stay dead for a while? And
With
then
what, with alcohol or?
With just the soap.
Well, it's just like soap and water. You're going to clean everything off, and so the soap breaks breaks apart the bacterial cells, or causes the viruses to slide off, and then everything's gone. But then stuff will start coming back in, and as long as it repopulates with your own bacteria, you're fine. The problem with an antibacterial soap is that nothing can repopulate, can grow, it will die as soon as it moves in there. And, you know, the odds are that you'll get the longer you have a dead zone, it's easier to find some sort of pathogen that didn't find a home before, can find one now as it eventually wears away. So, and that's what happens. It's easier to use an alcohol gel, like a hand sanitizer, kills everything for 20 minutes, then it repopulates. You have your own bacteria. It doesn't, most of us don't scrub our arms all the way up to the shoulder. We don't have a dead zone there for very long, and our bacteria come in. Soap and water does a very good, plain, ordinary soap and water does a really good job. It's one of those things, hey, let's put antibacterial agents in the soap. Sounds like a great idea. And in some cases, it probably is a good idea. But for daily activities, not so good. Our second line of defense consists of proteins in our body that are antimicrobial, meaning they're antibacteria, antivirus. Remember, viruses are not alive. Viruses are just a strand of DNA or RNA surrounded by a coat of protein, and they are coded to reproduce, and that's all they do. They don't particularly, they're not a living structure. They want to take over. They're programmed to take over a host. The last thing that they are programmed to do is kill the host. They're not programmed to kill the host, but usually if they reproduce sufficiently, they can cause the host to die, and that doesn't help them at all. So we have lots of white cells. Those are the phagocytes. We have numerous other cells that go after pathogens for us. We have a mechanism known as inflammation. Inflammation, the redness around a cut or a scratch, is a good thing for us. We believe it or not, we like to see inflammation. So now, so we have in our first two lines of defense, the are found in the non-specific mechanisms. Those are what we call the innate, meaning it's always there. Our skin, our mucous membranes. We have also the natural defenses, the phagocytes, what we call natural killer cells that are non-specific white cells that are going to go kill cancer cells. Inflammation. Inflammation is a good thing. We have the antimicrobial proteins, and we also have fevers. A fever is a good thing. A low-grade fever is our body's response to bacterial infections. It's a way to destroy the bacteria. We like that. We don't feel good, but we like the low-grade fever is not a bad thing at all. Now, the adaptive defenses, which we'll get to, deal with what we call the B cells and the T cells. We have what's known as humoral immunity. Those are the B cells. B cells produce antibodies. B cells produce antibodies. T cells go out and kill things. They give you cellular immunity. So these are the two types of white cells that our immune system, we call it adaptive. They learn to recognize foreign pathogens. This is why when you get a vaccination, the vaccination triggers a response of production of antibodies to go out and kill or destroy a virus or kill a bacteria. We've all had vaccinations for different things, flu shots, COVID, maybe, chickenpox, mumps, measles, all these. We've had a variety of vaccinations over the years. If you're old enough, or maybe your parents have mentioned being vaccinated for smallpox when they were a child. At some point, many of us were vaccinated for polio, which is something that is not a big deal anymore. So those are all part of the adaptive defenses. Now let's talk about, though, I already mentioned the surface barriers here. The skin protects us. It is it cannot be penetrated. Here, I got around it that way. It has keratin in there that makes it waterproof. The oils on the surface have antimicrobial properties. There are lysozymes in the saliva, enzymes to break in the saliva and the mucus and in our eyes to break down bacteria. There's all sorts, it's a physical barrier, it's a chemical barrier in here. So our mucus has antimicrobial proteins, peptides, that are going to restrict bacteria from growing any kind of bacteria. So we have all these different chemicals embedded in our skin and on the surface of our skin that'll trap the pathogen. If it's sticky, it gets stuck in there and it can't move and it's destroyed. And of course, we've talked about this many times about the cilia in the airway. The cilia and the mucus that trap bacteria and dust and dirt and stuff like that, which we sweep down into the stomach and we destroy it in the stomach acids. Now, any kind of penetration of the surface barriers of the skin of the mucous membrane lead us into the second line of defense. So this is still all part of the what we call the non-specific or innate, meaning the built-in. Our non-specific barriers protect us against all general pathogens. The specific mechanisms deal with individual pathogens, like you've been vaccinated against COVID. That vaccine doesn't work against chickenpox. You've been vaccinated against chickenpox. That doesn't work against measles. You've been vaccinated against measles. That doesn't work on polio. The polio vaccine doesn't work on mumps. Those are specific mechanisms. You get a sore throat from a rhinovirus. You have an infection. It's a sinus infection, and it's viral. It's not bacterial. It's viral. You get sick. You are sick, symptomatic for about a week and a half, and then you get better. That virus can never infect you again because you have your own antibodies to attack it next time they see it. You aren't even symptomatic. You have immunity. That's a specific immunity. We develop immunity as we go through life. Every time we're exposed to a pathogen, we get sick, we present symptoms, and then we get better. So while our first line, the non-specific is general against any kind of pathogen, the specific ones depend on having a response, having an antibody and T cell response. So let's talk about what happens when we get through the first layer. Okay, we get through this layer. Here's our trachea. And trachea is there to protect us. It has mucus. It's covered. Mucus is covering the cilia. The cilia are trapping all the dust and dirt. The idea is that the air that we breathe in is sterile. That's what we want. We want sterile air to breathe in. We want more moist, clean air. And in most cases, it pretty much is warm, moist, and clean. If you look at the lungs, you can see, the lobes of the lungs are going to trap dust and dirt particles that actually manage to make their way through there, and you'll see where it accumulates. Because some dust and dirt will eventually get through. It's heavier than the pathogens are. Now, you look at a smoker's lung. Smokers have paralyzed their cilia that we see here. See where the cilia are, where it says the luminal surface. The cilia are paralyzed. Their lungs are covered with all sorts of debris, all sorts of dust and dirt particles. Their lungs are much, much darker because of all the debris in them than a healthy lung. So here we have our second line. We've gotten through the first line. We have these phagocytes. We have natural killer cells. We have the inflammatory response. We have what are known as the antimicrobial proteins, which we call it interferon or complement. And then we have the fever. So what are the phagocytes? They're white cells. They're the neutrophils. They're the N in never let monkeys eat bananas. So the neutrophils, when they are exposed to some sort of pathogen, a virus, a bacteria, a fungus, they start, these neutrophils, the most abundant white cells, will turn into a phagocyte and they will start attacking the pathogen and try to destroy it. We have many neutrophils, and so we go after these pathogens and try and kill them. Yeah, they don't live long, but they will reproduce quickly. So they'll make more. We'll keep making them and keep making them and keep making them. Now the other type of phagocyte are the macrophages. They come from the monocytes. That's the M of never let monkeys. Those are the ones that look like big kidney beans. You know, that the nucleus has a big kidney bean shape. They are the number one phagocyte in our bodies. And so if we're exposed to a pathogen, the neutrophils will strike first, trying to go after the pathogen, and then we'll start spinning up the macrophages. Remember that, you know, in the order, it's neutrophils, lymphocytes, macrophages. You know, probably 90% or more of our cells are going to be either neutrophils or lymphocytes. So, you know, monocytes are a pretty low number here, but give them a little time to reproduce, to be produced, and we'll get enough macrophages to fight off an infection. Now we have two types. We have the ones that roam freely, looking for trouble. I'm looking for something I want to kill. You know, they're just like in the old Westerns, they'd be like the gunfighter that goes from town to town looking for somebody to kill. You know, that's what the macrophages do. The macrophages in our lungs are free macrophages. The fixed macrophages stay in place. We have them in the liver and the microglial cells in the nervous system. Remember back to AP1, microglia, one of the 5 types of glial cells in the central nervous system. They are monocytes essentially that are stationary in the brain, in the central nervous system, to protect us against pathogens that show up there. So these are, but they're non-specific. They'll attack any foreign structure, anything they recognize as foreign. And that also means good things like transplanted tissue. They won't care. What they're going to respond to is that they're seeing something with a foreign antigen on there. The identifier, the identifier that makes itself, the glycocalyc structure that sticks up and says, hey, I'm Bob's liver, but what am I doing in Fred's body? You know, type of thing. And so the macrophages are going to see that, it's foreign, let's go kill it. You know, so that's what they do. So this is what they do. This is how they work. This is a macrophage going after a bacteria. The pathogen, what you're seeing here, there's the bacteria, and the macrophage is going to engulf it. It's going to go completely engulf it like a big amoeba, and then it's going to destroy it. And you can see there's the bacteria. It looks like looks like rod-shaped bacteria. Bacteria only come in three shapes: rod or spiral or circular. And this looks like a cluster of rod-shaped bacteria. And the macrophage, the white cell, destroys it So, and we get rid of it. Doesn't matter what kind of bacteria it is. We destroy it. So we identify it as foreign by its glycocalyx structure. Remember the glycocalyx, the sugar-coated protein, the carbohydrate protein that sticks up on the surface of all of our cells that are the markers that identify it as us. This is us, this is my cell. Hi, I'm Bill's cell, that type of thing. And the white and all of our macrophages that recognize our cells, but you get a foreign cell in there and it says, we got to kill it, it's foreign. So, and again, that's the whole basis of rejecting tissue and transplants. You put in an organ from somebody else with their antigens on the surface of their kidney. Well, your macrophages are going to be saying, hey, that's not us. And they're going to attack it. Okay, now, natural killer cells are a different type of white cell. So we've seen the neutrophils and we've seen the monocytes as the phagocytes. We've seen neutrophils and monocytes. Monocytes are the most common. They become the macrophages for us. Now what's going on here is the natural killer cells are lymphocytes. Lymphocytes that monitor our blood and our lymph. They like to hang out in the lymph nodes. And they're going to kill cancer cells. They're going to destroy cells that are infected with viruses before the adaptive immunity, the third level, kicks in. Natural killer cells that what they see, they look at foreign cells. They say it's a foreign cell because it doesn't have an antigen sticking up. So these are cells of our immune system, whether they're specific or non-specific, whether they are these innate, naturally occurring protection, they're looking for cells that don't have any markers, any antigens on there. And they say, well, that's not right. Everybody has an antigen. It's like driving without your license. That's not a good thing. Okay, so these bacteria and these viruses are out there without any antigens, and the white cells detect that. The lymphocytes detect that, and it's usually the cancer cells and cells that are infected with a virus, because the cancer cells, they're changing their antigen. They keep changing and keep changing and keep changing their antigen, which is why our immune system has a real hard time with fighting cancer cells off. How do you attack something that keeps changing the target? Our adaptive, our specific immune system, like the one that protects us with a vaccine, for example, it takes about 2 weeks to spin up. So you have a cancer cell with a foreign antigen on there that your immune system is going to recognize as foreign, and it's going to say, okay, well, I see that you're a cancer cell, and I recognize your antigen as a foreign substance, and I'm going to produce a whole bunch of these white cells that are going to come and kill you. And by the time they produce that whole bunch, that's a technical term, by the way, whole bunch. Okay, whatever. A lot, millions and millions of white cells. By the time they've done that, the cancer cell says, whoops, I just changed my marker. I'm not who you thought I was, Oh no, that was them over there. That's not us. See, look, we have a different antigen. And so our immune system can't go after it. Our specific immune system can't go after it, but our non-specific immune system doesn't care. It'll kill anything like that. And it will trigger apoptosis. It will trigger the cell to destroy itself. Any cell that does not have an antigen on it, any cell that doesn't have the right receptors on it, are targets for destruction. Now, can they win the fight? Sometimes. We don't know how often we're exposed to cancer cells. We don't know how often cells may become cancerous that our body's immune system takes care of. It may happen all the time. You know, to develop a cancer requires a lot of things to happen at, you know, essentially at the wrong place at the wrong time. You know, and it's not something that you know, how can I say this? If cancer were easy to acquire, we'd have a lot more people developing cancer. Let's put it that way. So a lot of things have to go wrong in the right order for cancers to develop. So our natural killer cells are protecting us from these various cells that may become cancerous or are infected by a virus. And so we don't develop cancers. You know, I mean, what is the statistic? Was it 1 in 8 people will develop cancer in their lifetime? You know, and again, how come? If you take away all the risk factors that you know, your person smokes, your person is exposed to toxic chemicals, your person is exposed to HPV without being vaccinated for it. You know, you take away all the things that are cancer-causing agents, that number of, you know, cases probably drops to like 1 in 20. Just saying here. Now, okay. Natural killer cells kill cancer cells and cells that are infected with viruses. They are not specific. They'll attack anything. That's what you need to remember. Now, interferon is an interesting protein here. It was thought years ago that interferon was going to be a great anti-cancer drug. What interferon is, it's a protein produced by a cell infected by a virus. So we have an infected cell, and the cell is infected and it starts producing interferon. Now the interferon doesn't help that cell. That cell is now infected. It will probably die. But the interferon is released to the cells around it. The interferon is absorbed by the cells around it and starts blocking all the receptor sites for a virus. So you have an infected cell, it produces interferon, the interferon goes out to all the cells around that are healthy, and the healthy cells absorb the interferon, and now when the virus tries to infect the healthy cell, they can't, because the interferon is blocking. Because the virus will only land at can only land at certain locations on a cell. It isn't like it can just come up to any cell and say, hey, hi, here I am, you know, I'm going to infect you. It has to have certain locations to do that. And those locations are blocked by the presence of the interferon. The cell doesn't become infected. The cell doesn't become symptomatic. The cell doesn't make more viral particles. That's all a virus wants to do. It wants to infect the cell and get the cell to produce more viral particles. And it will keep doing that until the cell will make copies of the virus and copies of the virus. And then the cell explodes and releases those viral particles out into the body of the host, which will then infect other cells and other cells and other cells. Now, the problem is, how rapidly can it work? Some viruses kill within 2 or 3 days. You know, Ebola virus has a mortality rate of about 90%. It kills this, it infects the body's organs faster than they can recover. If you could slow down the rate of viral growth in an individual exposed to Ebola virus, for example, it would give the body's own system time to fight back. But it kills so rapidly that the infected person's immune system can't fight back in time, and they die. And you know, people that were exposed to COVID and tested positive for COVID, but never showed any symptoms and got better, their immune system was able to attack the COVID virus and destroy the infected and keep the infected cells from reproducing rapidly so that it couldn't spread. And so they were barely symptomatic. You know, they may have had sniffles, they may have had a sore throat, they may have had a cough, they may have had none of that and still tested positive, simply because their immune system was robust enough to stop the virus in its tracks. This is what interferon does to us. Interferon prevents other cells from being infected. It also then can prevent any viral particles that have been formed from infecting other cells too. So this is sort of a, it's sort of like the last great altruistic behavior of, I'm going to sacrifice myself for all the other cells around me. You know, so, and that's what's going on here. So this is one of the, this is a non-specific way when a virus infects us that we, just because we're infected by a virus, remember this, just because we're infected doesn't mean that we are going to have any symptoms, because we can be infected, we can be exposed to a virus all the time and never show symptoms. Perhaps our interferon response is so powerful that the virus doesn't get a chance to do anything. So this is, you know, it's non-specific. You're exposed, you know, you haven't been vaccinated against measles, mumps, or rubella, or anything else, and yet you have never been sick a day in your life. Maybe your interferon is stopping those viruses in their tracks. So now complement is another protein. When a cell is infected with a pathogen, it can be a bacteria, it can be a virus, it will produce complement that will start destroying the bacteria. It will start producing it will actually enable itself to punch holes in the membrane of the bacteria. Remember, bacteria are living, they have a cell membrane, and with complement, it can be used to punch holes in the surface of the bacterial membrane, and cells don't like having holes stuck in their side because water leaks in and sugars leak out and bacteria tend to pop, you know, sort of like that balloon the other day. So when you jumped,
I went to a surprise party after that.
Uh-huh.
With friends, and then when we were cleaning up, there was like a bajillion balloons, and they were stabbing them with scissors, and I was freaking out. So thanks for that. It's fine.
Hey, I just, you know, prepared you for.
Prepared you for that.
Yes, thank you, Abbott. You were prepared.
Yeah.
My wife hates
I
balloons.
got cried. It
She,
was so loud.
the only balloons that she likes are the Mylar balloons because they can't pop. They just, you know,
Exactly.
she hates those kind of
The
balloons.
latex ones. or
Yeah.
whatever it is.
Yeah, they're latex. Yeah, she hates those. Her idea of a good balloon is one that the string's been cut and there it goes, floating away. You know, I think she just likes to see kids cry, but you know, no, not really. No. No, I didn't say that. I didn't say that. Now, here's what complement does. Complement punches holes in the surface of a bacterial membrane. It's beautiful the way it does. It comes up and it literally punches a hole and the water leaks out or water comes in and the cell explodes. The bacterial cell explodes. It also functions as it generates a chemotaxic response, meaning it sends out proteins from itself that attracts phagocytes to the bacteria. So it's a very elegant little process for getting rid of bacteria because, you know, there's a lot of pathogens out there and most of us go through our life without being sick a lot. You know, we all get sick occasionally, but it's not like with all the pathogens that are out there, we should be, you know, theoretically, we should be, you know, unhealthy every day, but most days are pretty good. You know, it's unusual to, you know, I would venture that most of us are a lot healthier than we were when we were kids, when we were exposed to everything. Yeah. See, so, you know, our immune system, both the first and second level and the specialized level, are much more powerful than they were when we were kids. So part of that is we were learning how to respond. Our bodies were learning how to respond then too.
That's why I think daycare is wonderful. Kids like they get exposed. My kids been at daycare since they were 6 months old. They were sick almost every other week until they were about 2. And now my daughter's in kindergarten. She never gets sick.
Oh, yeah,
because
yeah.
she's been exposed
know,
to everything under the sun.
You know, socialization with other kids is a great way to guarantee that they're going to be sick a lot early on, and then they're going to be better.
Mhm.
You know, the whole idea of not letting kids play outside or play in the dirt, or, you know, it all started with, well, windows that don't open. Windows that don't open. It all started with, you know, you were gonna,
Yeah.
you know. Oh, go ahead.
Go ahead and say it.
What? I want to see what you're gonna say. The what?
The C word. COVID.
Oh, well, it was, okay, COVID, but it was before
But now
COVID.
you're not allowed to do anything. They
No,
don't want you to
no.
touch nothing. They
No,
don't want
you
to do
know.
anything.
And I dare say that kids are probably paranoid, which is why lots of kids are afraid to take their mask off. You know, I think they're paranoid over, you know, young children are very impressionable, you know, and, you know, I think that there's a lot of fear going on out there. So,
I think that's why my kids never, I had it, my husband had it in the house, like when it first came out, and that neither of them got sick, but they're around where germs all the freak, they probably
Mhm.
had COVID.
oh, yeah.
And didn't know it because they didn't have any symptoms because they've been in daycare, you know, for forever, and they didn't get sick.
Mm-hmm.
They might have had it, didn't know, but they never
Yeah.
got sick though, like me and my husband did.
So now this is what complement does. This is your bacteria. The complement lands, the protein lands on the bacteria and punches holes in it, you know, and it's like driving over a nail, you know, which, you know, still flattens tires. You know, if you've never had a flat tire from driving over a spike or a nail, it'll happen given enough time. So, and what will happen with the bacteria is you punch holes in it, water comes in, and the bacteria pops. And if you do this enough, then the bacteria is dead and you're not symptomatic. And the most important response, I think, though, with all this is inflammation. When you injure yourself, and we do that all the time, you know, if you, we walk into things, we cut ourselves, we get paper cuts, we play with our dogs or our cats, and we get scratched, or you have a dog that's, you know, loves to jump with sharp claws, and, you know, yeah. So, well, I have 2 dogs. The Basset loves to come up behind my leg to get my attention because I wasn't looking at her, you know. So
We've been sitting in the house with no air conditioning for a little bit.
yeah. Oh, when I went home on Tuesday, it was the weather. It was very nice.
So they weren't too upset.
I mean, Monday they weren't upset at all. So, so anyway, the inflammatory response is how we deal with infections. Whenever we have a penetrating injury, you know, you have redness around the injury. And there's a reason for that. The redness is a good thing. The whole inflammatory response is designed to promote healing. And the 4 signs of the inflammatory response that's working is redness, heat, swelling, and a little bit of pain, or maybe a lot of pain, but it depends. So redness, heat, swelling, and pain. This is inflammation. The idea with inflammation, even in this case, which looks like there's a lot, you know, that's that redness around the top of the foot there. The inflammation is caused by increased blood flow. That's the whole idea behind it. Inflammation, the redness is showing that there is increased blood flow to the region. It's not necessarily a sign that something's bad. I mean, I've got my latest marks on my hand are all, you know, little red areas. You know, we probably all had something like that. What happens there is the inflammatory response triggers increased blood flow to an area that's been damaged. So the trigger is histamine, the same histamine that causes our noses to run and our eyes to water, which is also an inflammatory response. Histamine causes our blood vessels to dilate. It causes the capillaries to become more permeable. So our blood vessels dilate, the arterioles dilate, the capillaries become more permeable, and we have water leaking out of the capillaries. Okay, so we got more blood flow. You know, histamine comes out, blood vessels dilate, more blood comes to the area of the injury. That gives you the redness. It's red because there's more blood going in there. That's as simple as that. We have swelling because the blood vessels have dilated, which causes the increased blood flow and redness, but it also causes the capillaries to become more permeable and more plasma leaks out into the tissues. Now, why is it, you know, there's a reason for that. Plasma is leaking out. White cells can follow the plasma because white cells leak out of can move out of the capillaries through diapedesis. They can squeeze out through those little tiny openings in the capillaries. Well, they follow the plasma. So now you've got increased blood flow to a region, and you've got plasma leaking out into the area, bringing white cells there. Well, when you have more blood going to an area, you're going to be bringing more oxygen and more glucose. So here you have your cut on your skin. It's turning red. More blood flow coming to that region. Swelling, because plasma is leaking out of the blood into the interstitial spaces around the injury. We swell because that we can't compress fluids. The swelling occurs from plasma leaking out there in the space. We call it edema. It's red because of increased blood flow. It's hot. We feel heat from the area because we've got more blood going there, and blood is hot. Blood is 98, it's like about 99 or 100 degrees Fahrenheit. It's a little bit warmer than body temperature. So the cells that are damaged are going to release histamine. They're also going to release heparin. Heparin's an anticoagulant. We don't want clotting here when we are trying to bring in more blood. Now, we will clot around the site of the injury, but we're not going to clot and block the flow at all, the complete flow. We want a lot of blood to come into the injury because we want to bring in white cells to kill bacteria. We want to bring in glucose so we can promote cell growth for repair. We're going to bring in oxygen, again, also to promote cell growth. And so we get redness, we get heat because we've got, you know, more blood coming in. It's red, it's hot because we've got blood's coming in there. We have swelling because the plasma is leaking out of the capillaries, and we have pain, a little pain, because we're filling up the spaces where the water shouldn't really be, and it's pressing against pain receptors. So it's tender, it hurts, you know. At this point, you know, all I've got, nothing on this arm, but.
I got one on my forehead. My son threw a Donkey Kong cart. It
Okay,
hurts.
okay, I see it, yeah.
Yeah,
I'm sorry for that.
it's
Oh,
bad.
So you're not gonna get extra credit for that, by the way.
Yeah.
Yeah, no, sorry, you know.
Looks like a baseball on my elbow.
Oh,
Like the stitches on
okay.
a baseball.
So the pain is caused by the swelling of the fluids. You know, you're having an inflammatory response, which is a good thing to cause it to heal.
Yes.
Your inflammatory response, all of our inflammatory responses are promoting healing. That's what they do. And they hurt to various degrees because of the pressure by the plasma that leaks out. That gives us the pain. You know,
I said it definitely does hurt.
well, you know, but you are experiencing the joys of an inflammatory response.
I don't know about yours.
Oh, in a couple of days, it'll feel better.
I mean, it's hard to feel better.
Yeah.
Yesterday,
Now, you know, did they tell you to take any anti-inflammatories like Advil
just-
if you need them? So again, they work through,
Taking a prescription, I had to have They gave me hydrocodone and I didn't take any of it.
That's probably a good thing not to take that. So, yeah, so.
I didn't want to.
Okay, now, so this summarizes the whole thing for us. We have a penetrating injury into the skin. We generate the release of histamines, which causes the blood vessel, the vessels to dilate. We release heparin so that we don't clot. We bring in more blood, which brings in more glucose and more oxygen. The plasma leaks out at greater, through the openings in the capillaries, and that gives the white cells a way to get out to the site of the infection and kill the bacteria that may have come in, that did come in. So, and so it gets better. One of the beauties of is that when we have a cut, we don't have to do anything. It's going to heal up on its own. The healing process in your arm, the healing process here, and on my cuts and scratches from my dogs, I don't have to do a thing to heal up. So, now, if we want to have the proper terms for it, we have the phagocyte response here. We have leukocytosis. We increase the presence of the neutrophils. We bring in the presence of the monocytes. We have the white cells will migrate to the area of where the injury is located, and then through diapedesis, they will squeeze through the capillary walls and start attacking any pathogens that may be there, or getting rid of damaged tissue. That's what they do. That's the great thing that they do for us. And again, this is a real slow animation, but you can see the white cells are coming out of the blood. That's the diapedesis taking place, the neutrophils and the monocytes. And there's our penetrating injury with our bacteria. There's bacteria on everything. Everything. There is no such thing as a 5-second rule.
Unless it's a kid.
Unless it's a kid, or it's something that you really, really like, yeah. If it's shrimp,
A French fry.
or frog legs,
No risk.
if it's something that's like, is that last piece of steak,
Yeah.
there is no 5-second rule, if your favorite dessert falls on the floor, but really there is no 5-second rule. Everything when it hits the ground, hits any surface, is contaminated. It's contaminated though when it hits the plate and you put it on the plate to eat it.
So you'll live.
Yeah, so people haven't died for thousands of years from eating stuff off the floor, but it's probably not a good idea. So, okay, now and let's see what else. Okay, here we go. Oh yeah, let's get the fever. When we get infected, when we have a bacterial infection, our hypothalamus will often reset our body's temperature. And we don't like high temperatures, 103, 104, those are dangerous. But if we have a very moderate fever, like 99, 100, something like that, this is a good thing for us. When the hypothalamus raises our temperature just a little bit, it causes us to start retaining zinc. Our liver and spleen retain zinc and iron. Bacteria need zinc and iron to reproduce. If we suck it all up in our spleen and in our liver, then the bacteria are going to be inhibited. We also, if we to get our temperature to go up, the hypothalamus increases our metabolic rate. The higher the metabolic rate, the faster our cells heal and kill pathogens. So a low-grade fever is not a bad thing, despite the fact that we feel uncomfortable when it's going on. So we don't like high fevers. we don't like extremes in the temperature ranges either way, because we're going to destroy the enzymes either way with this. So, but we do, while we may not enjoy it, a low-grade fever is not a bad thing. So,
They told me after surgery, they're like, if you have a fever, as long as it's not above 101, you're okay. Like, they said, I never had a fever, but they were like, it's possible that you will have one. But they said if it's anything, like, if it is above 101, I have to go to the ER. And I had to go, but not for fever.
well, and well, that's good that you didn't have the fever. A higher fever indicates that the bacteria are growing at a faster rate. You know, our
Just worried about like infection.
yeah, the sign infection, a infection that's growing is going to trigger the higher fever, and so you don't want to see that. Low grade fever, fine. You know, we've all had low grade fevers, and we may have experienced high fevers too at some point or another. When we were kids, we probably all had something like that. So, okay, and that is everything I want to talk about on the non-specific immune system. Now, let me get out of this. Unfortunately, we still have some time to go into the specific system. So, and let me get out of this. There we go. Okay, now our third line of defense is specific. It is tailored to each type of pathogen that we encounter. Whereas the non-specific mechanism will respond to anything. Any pathogen that comes along, it'll try to attack it, destroy it, and kill it. That's what it does. It doesn't care, but it's non-specific. The specific mechanisms are tailored to each individual pathogen. As we become adults and as we age, our memory of foreign pathogens gets larger and larger and larger. We can respond much quicker to pathogens that we get exposed to, that we've been exposed to in the past. We can respond much quicker than we did the first time around. You know, if you had chicken pox, or if you were exposed, or you had the virus, the vaccine. If you had the chicken pox vaccine, or you had chicken pox, usually you don't get chicken pox again if you had it once. Now, I used to say, with 100% absolute certainty and conviction, if you had chicken pox once, you never got it again. And I started getting people raising their hand and saying, well, I've had it twice, or I've had it 3 times, which means that you can't always depend on your natural immunity, to protect you the second time around. Some people have had it twice, some people have had it 3 times. They haven't got it past that. It all depends on what kind of response we generate when we're exposed to a pathogen. But in general, most people, when they have a disease, are immune to it from then on out. Most people, when they're vaccinated, are immune to it from then on out, except COVID. COVID doesn't seem to, vaccines aren't going, the COVID vaccines were told to us that they would protect us from COVID. And so you take the vaccine and someone gets COVID 6 months later.
It's usually the ones who have a vaccine have noticed that.
Yeah, I've noticed that too.
Yeah.
So it's not that you're never exposed to those pathogens again when you get a flu shot, so you don't get the flu. Okay, you get the measles vaccination, you don't get measles. It doesn't mean you're never exposed to them. You're probably exposed to them all the time. You've got kids, and they go to daycare, and that's just a giant petri dish of bacteria.
Like your wife.
Yeah, exactly. But I will say, since she teaches at New Center and not at Sevierville Middle, she doesn't get sick near as much.
Really?
I suspect it's something in the AC system
Really?
there for here. Yeah. So I don't know what the difference is, but I know that.
It's smaller than two, isn't
Yeah.
it?
Well, yeah. Which one? New Center?
Yeah.
Yeah, it's a smaller school,
So there's
and they're broken up in the smaller units. Whereas at cerebral, middle, and intermediate together like that,
Yeah.
there's a lot more opportunity for pathogens to spread. And there's more people there. And then you got Trula right behind them, and there's interaction across there. So you have a much larger pool of bacteria to come in there. And you got different ages of the air conditioning system. You got some new areas, you got some very old areas. And so, I've often wondered, are those systems as efficient as they should be? But I know that since she's been at New Center, she doesn't get sick very often. Of course, it could be
Because she's been
yeah,
exposed.
she's been exposed at cerebral middle.
Yeah,
So now she goes to New Center and she doesn't get sick.
yeah.
So she was vaccinated,
Mhm.
so, and that's probably,
I feel like that's the best vaccine, is being exposed to
Oh,
stuff
yeah.
directly, not the
Yeah.
actual vaccine.
so,
Yeah.
but we go through life exposed to these pathogens all the time. We don't become symptomatic because we mount an immune response right away. Whereas the first time we're exposed to it, we get sick. The second time, we're not even aware of it because our immune reaction, our specific immune reaction is so rapid. We don't even know. The only way that we would know that we were exposed to a pathogen is to have blood work drawn and see if we have antibodies to chickenpox rising in our body, because that's how we, if we're producing antibodies to the chickenpox virus, for example, that tells us that we're responding to the presence of the virus and we're destroying the cells that are infected. We're not even aware of that. So, okay, what the adaptive specific mechanism does is it will recognize a foreign antigen, a virus or a bacteria. It will destroy the virus. It will kill the bacteria or block its action. And it will trigger, to help promote the healing process, it can trigger the inflammatory response. It can activate complement to punch holes in the bacteria. When you get the flu, one of the side effects of the flu, besides the coughing and the sneezing and the vomiting and the diarrhea, is often a fever. That's part of the whole healing process. So all these responses are going on here whenever you get sick. A lot of the illness part of the flu is your body fighting off the virus. So now, okay, so the adaptive defenses depend on the B cells and the T cells. The B cells produce antibodies. The T cells are killer cells. So we call it humoral immunity. Humoral immunity is provided by the B cells. Remember, these are lymphocytes. Remember, we have never let, the L of never let monkeys are the lymphocytes. Right, neutrophils are the most common. Lymphocytes are the second most common. They represent about 30 to 35% of all white cells. Neutrophils are about 55 to 60%. So between the two of them, they're well over 90% of all the white cells. So T cells are going to be cellular immunity. The third line of defense, the adaptive immune system, is antigen specific. It's body-wide and it has memory. We retain memory of every pathogen we've ever been exposed to in our adaptive immune system. Whether the pathogen was a virus or it was an infection. Whether it was a virus itself or a bacterial infection, or it was a vaccine to it. So we remember, we have memory. Years ago, in the 1700s, individuals would inoculate themselves with smallpox from a person who had a very mild case of it, so that they would get smallpox and then they would be immune from then on out. It didn't mean they were never exposed to smallpox. It meant that their immune system protected them. At some point, the idea was taken one step further when it was discovered that if you infected a person with cowpox, a very mild infection, smallpox had a mortality rate of about 40%, and if nothing else, it would cause these lesions on your face like chickenpox does, only these would be deep pock marks, like someone had gouged out the skin, and it would be disfiguring. So if you survived, you might survive and be just fine. You might survive and have all these scoop marks on your face, or you die. You have a 40% chance of dying. So, but cowpox it was sort of like chickenpox. It produced 1 or 2 lesions, if any, but then you were immune to smallpox too. So the idea was that you would trigger a response. The antigen on cowpox and the antigen on smallpox were so close that our immune system could recognize it as almost the same thing. And so our response would be the same. Our memory to exposure to an antigen will last a lifetime. The shots that we got as kids for measles, mumps, and rubella, and for diphtheria and pertussis and all those other things that we were vaccinated for. The reason that our immune system, the reason that we never got those diseases is because we had memory of all those. Our immune system retained a memory. So the humoral immunity deals with the production of antibodies. The cellular immunity is the immunity conveyed by the T cells. The B cells produce antibodies. I guess you remember B for antibody, and T cells are the cell mediated, the cells provided. So B cells and T cells, but the key thing here is that it's memory based. T lymphocytes do not produce antibodies. B lymphocytes produce antibodies. A third cell that comes in here are what are known as antigen presenting cells. They will bring antigen fragments to the presence to the white cells to say, hey, look what I found. There's some sort of foreign antigen out there. We need you to do something. Okay. T cells and B cells, they're both produced in the bone marrow. Remember, lymphocytes are blood cells, and blood cells are made in the red bone marrow. T cells mature in the thymus gland. Remember, they all go to our T cells, migrate to our thymus gland in the first year of life, and they are trained to become immunocompetent so that they can recognize foreign antigens. The T cells can recognize foreign antigens here. One of the reasons that in males that the testes are contained in the scrotum and have outside the body cavity in the male is to keep the T cells and the sperm cells from interacting. Sperm cells didn't start forming until the male's about 10, 11, 12 years old, whenever puberty starts. They weren't there. Their antigens weren't present. Their glycocalyc structures on the sperm cells weren't there in the first year of life for the T cells that were becoming immunocompetent to say, hey, those are us. Don't destroy them. T cells in the thymus gland recognize the liver, the spleen, the bones, other blood cells. They recognize the small intestine, the large intestine, the kidneys. They recognize every structure in the male body except sperm cells, because they weren't there yet. They weren't there. They won't be there for 10 years. And by that time, the T cells have already become immunocompetent to every foreign structure, every structure in the body. In females, the ovaries already have the eggs. So their antigens are already recognized by the T cells. So the T cells become immunocompetent in the thymus gland. The B cells become immunocompetent in the bone marrow. They recognize then, immunocompetent means their ability to recognize foreign antigens. Remember, the antigen is the glycocalyx that identifies all of our cells as self. T cells. Okay, I just said most of this already. They mature in the thymus. Any T cells that can't recognize foreign cells, any T cell that persists in trying to attack our own cells is eliminated. They get rid of it. The only T cells that will recognize foreign antigens are kept. Now, our B cells and T cells, once they become immunocompetent, then they generally spend their time in the lymph nodes in our bodies. And what better location? The lymph nodes receive lymphatic fluid from draining back to our blood. Lymphatic fluid is nothing but plasma that has leaked out into the interstitial spaces where it becomes interstitial fluid. It's collected in the lymphatic vessels, and then it drains back to the plasma. It passes through lymph node, these lymphatic organs called lymph nodes. We all know about lymph nodes. We have axillary lymph nodes, for example, and our T cells and B cells hang out in the lymph nodes looking for foreign substances that shouldn't be there, but so they're watching. T cells are looking for something to kill. That's what they do. The B cells are looking for something to produce antibodies to. The B cells function differently than the T cells. The T cells are the only cells that will kill things in our adaptive response here. The B cells are going to produce antibodies. Now, immunity is something we don't have when we're born. We have no immune system. We have borrowed our mother's immunity, and that lasts for about a year. So we in that first year of life is when we start getting all of our shots. Now, immunity is acquired. We acquire our immunity. We acquire immunity to a specific pathogen if we've been exposed to that pathogen, or if we've been exposed to the antigen on the pathogen. This is what we call acquired immunity. If you get a vaccination, you get a flu shot, they don't inject the flu virus into you. That would be unpleasant. What they do is they inject in the antigens from the protein coat that surrounds the flu virus, because that's what's going to trigger your immune system. So you get a shot in October for influenza, and what you're getting is nothing but the antigens, the glycocalyx structures on the outside coat of the virus. Your immune system recognizes that as a foreign antigen, mounts a response, and it stores that response in memory. So every time you're exposed to the flu throughout the course of the winter, which you have been, if you go to Walmart, you've been exposed to it. If you go to Tanger, you've been exposed to it. If you go anywhere, you've been exposed to it. The flu virus particles are everywhere, theoretically. But you don't get sick because you've already been exposed to the antigens. The antigens triggered an immune response in your body, and now that response is stored in memory. This is acquired immunity. We acquire immunity by being exposed to a pathogen or to the antigen from the pathogen. Now, acquired immunity is either active or passive. Yeah.
Not to backtrack or anything, but, my grandpa was just diagnosed with non-Hodgkin's lymphoma,
Uh-huh.
and it's not in his lymph nodes or anything, but it's in his bone marrow. So that's not good for it.
It's, I don't want to say it's not good or, it's,
Like, he's 81. I don't think they're gonna do, if they did a bone marrow transplant, would that even help or not?
if you get rid of the bone marrow, then you get rid of the condition. So,
He's had 2 treatments so far.
how's he recovering, doing from the treatments?
He was a little, he felt a little sick.
Mm-hmm.
For like 2 days, he could barely taste things, and he was like nauseous and like tired, but he feels fine now.
Mm-hmm.
He had the treatments on Wednesday and Thursday last week, was sick Saturday and Sunday, and now he's fine.
Okay. It's hard to say, every,
He said I didn't understand it.
Yeah.
Because they said it's good that it's not in his lymph nodes and his spleen,
Mhm.
because they thought it was at first, but it's not, so I guess that's good. But now they're saying that it's in his bone.
Well, if it's in his bone marrow, it's less likely to spread, I would think,
Yeah, that's what.
because the lymph nodes, it gets in the lymph nodes, it spreads everywhere.
Yeah, and they said that since it's not in his lymph nodes or spleen now, and it's not gonna, now
Yeah.
that they started treatment.
Yeah, so they can stop it now. It's sort of like, it's almost like having it cut out, where, you sort of localize where it's going to be. So,
That makes sense.
so try to keep me posted on that.
Yeah, I don't know a lot.
So
Like, they just. Sorry.
that's okay. Now, I'm going to have to wrap this up. Things to remember about immunity is that B cells and T cells are going to ignore our own antigens, our own glycocalyc structures, the structures in all of our cells. They're only going to go after foreign antigens. And the first time they do that, they always generate memory. So that we can, every time we're exposed to that same pathogen again, we can mount a response. The thing about the first time we're exposed to a pathogen, let's say that you get the flu. Okay, you're exposed to the flu. It takes you about 10 days to get over. Because it takes that long to mount a response to produce enough antibodies to isolate the foreign antigens. And you either isolate them and target them for destruction, or isolate them and block them from acting. Because flu virus wants to infect other cells. If you block it from acting, then it can't infect the cells. And eventually you're going to start, your T cells are going to start destroying the infected cells, and your antibodies are blocking the virus, and you get better. It takes about 10 days. If the second time you're exposed to the flu, after the first time, you have a memory of that particular antigen. Let's say it's the antigen named Bob. So the next time Bob infects you, you don't wait 10 days. Instead of taking 10 days to build up your response, your response is in about 10 minutes. And so you crush the virus, so that you're not symptomatic. And then the next time, and the third time, and the fourth time, and so on after that. So there is a lag. The first time you're exposed to it on what we call the primary response. The old joke from physicians will tell you, because there aren't a lot of antiviral medications for the flu. There are some, but not many, because we generally treat the symptoms and let you get over it, or you get a flu shot. They say, well, if I don't give you anything, you're going to be sick for 2 weeks. If I don't treat it at all. But if I treat it, you'll only be sick for 14 days. Treat the symptoms. So anyway, what I'll do is I'll go ahead and record a separate part of this and post it for you, because there's just I could go on and on and on. It's an interesting topic, but so let me get out of here.
So once
And
you have it the second time, even though you're not symptomatic, you technically still have. flu.
yes. Even if you've had it you're,
Even though
you're not sick,
you still.
you still are, you still have the virus. The only way, or the pathogen, the only way you would know is to have a test to see if the antibodies are there. That's all. That's the only way you would know.
Yeah.
So,
Another question.
yeah.
Okay, so at first, when COVID first started happening, they were saying that like people who were asymptomatic couldn't spread it. And then people were like, they came back and said, if you do have COVID and you are symptomatic, you can, like, asymptomatic, you can still spread it. So like, is that, I don't know what,
Well,
I
okay,
don't understand.
you could be infected and not have any symptoms.
Yeah.
And so you're still carrying the virus inside you. And so.
So if you like, say you cough on somebody, but you don't have symptoms, you could still give it to
Right,
them?
right. You could feel perfectly fine. And, the only way you know is have a COVID test, say, oh, you tested positive, which is why they made people go quarantine.
Yeah, it's like, kind of like when people say if you don't have a fever, you're not contagious. Is that true?
Not really.
Yeah, I was gonna say, cause like,
Yeah.
that's the same thing as being asymptomatic.
Yeah, because if you don't have a fever,
You're still infected.
you're still infected. It just means you're not producing the fever. You may not, lots of us, so, many of us are infected and don't even know it. Because, if you're not showing any symptoms, how do you
How
know?
do you know?
Exactly. The COVID test was the only, the quick test for COVID was the only thing that we had recently to say, oh, you're infected. And, COVID is one of those things where they, not in a bad way, but they were making a lot up as it went along because we didn't know. We
They're
didn't
trying to give
know.
people answers.
Yeah, we're trying to give out answers for stuff that we didn't know,
And that's just as dangerous.
Yeah. So, and then it became politicized, and that just went, yeah, so,
Well, I'd love to say cough, but I'm going to a choir party.
okay, well, I have another class coming in here shortly, so.
I'm so sorry.
Okay, any questions from anybody before I shut this down?
Thank you.
This is the last class. The final opens, thank you for asking it. This is the last class. The final opens up Monday morning at 10:15. It will stay open for 24 hours. Take it anytime you want in that 24-hour window. If you have a conflict, let me know. If you take it at 10:15 on Monday when it's scheduled, you shouldn't have a conflict with any other class. The reason I'm giving everybody only a 24-hour window is I have to have grades in by Thursday. So I have a short time frame here. So, and thank you all for being here and putting up with me. So, and I will talk to you all later.